Alright, so now let's go to the next section of this paper. It's still on the paper methods, but it's going to be on the electron microscope preparation. Just basically the electron microscope is going to be done on the supernatant of these cultures and the overall cells over there, the bottom part. So anyways, now let's take a look at the next part of the paper, which is preparing the samples for electron microscopy. So transmission electron microscopy. A supernatant from human airway epithelial cell cultures that showed cytopathic effects was collected, and then also inactivated with 2% paraformaldehyde for at least 2 hours and ultracentrifuge to sediment virus particles. Alright, interesting. So virus inactivation refers to the process of removing, quote, viruses completely or rendering them, quote, non-infectious. And paraformaldehyde is a chemical preservative used to, quote, fix proteins or other molecules in the sample via cross-linking polymers, thus locking them in place and preventing any further changes. And fixation is the preservation of biological tissues from decay or degradation to make them suitable for subsequent analysis. And a cross-link right here is a bond or a short sequence of bonds that links one polymer chain to another. And the 2% paraformaldehyde indicates the concentration of the solution, so 2 grams of paraformaldehyde for every 100 milliliters of solution. And an ultracentrifuge, right here, because it's ultracentrifuge, basically a very fast centrifuge with speeds up to 100,000 RPM or revolutions per minute. And transmission electron microscopy, TEM, involves firing a beam of electrons through an ultra-thin section and the resulting interactions with the sample forms an image that can be made visible. Through various methods such as a fluorescent screen, and electron microscopes provide far greater zoom than optical microscopes, but the samples used are of dead material because of the high energy electron beam and typically stained with heavy metals. And heavy metals have relatively high densities or atomic masses, and this fluorescence right here, fluorescence is the emission of light by a substance that has absorbed electromagnetic radiation. And electrons and electromagnetic radiation, EMR, are not properly defined by mainstream science, according to my opinion, but a good way to view them is as interactions with an all-pervasive medium, the ether or the Aether. Electrons can be viewed as vibrations in the ether, and EMR, electromagnetic radiation, as the resulting waves in the ether. So you could think of, electrons as vibrations. They have an up and down like this, just like a puddle, and then the radiation is the resulting waves that radiate from that vibration source in the ether. And some good resources on this are Einol, Dino, and Fractal Woman YouTube channels right here. I put the links on there, so check those out. All right, then going further, so here's an image of this transmission electron microscopy. So transmission electron microscope operating principle, basically you would fire a beam of electrons, and then focus it down to the sample, and then the interactions through it, you would focus it back onto this right here, to this fluorescent imaging right here, and you're going to see some dots in there, and this would represent these different grooves and so on of the sample. And then this is a fluorescent plate right there. So the beam is then magnified by other lenses and detected, for example, using fluorescence right here, fluorescence screen. Fluorescence just emits light by a substance that has absorbed electromagnetic radiation. So it absorbs it unevenly, then it's going to reflect, and then it's going to reflect visible light out, or emit visible light out. Then you can see the differences in the contrast and so on. That's very interesting. All right, continuing further in the paper, the enriched supernatant was negatively stained on film-coated grids for examination. So enriched supernatant refers to the portion of the centrifuge supernatant that contains the sedimented, quote, virus particles, and the term enriched refers to the concentration of the solute, in this case the, quote, virus particles, has increased. So yes, it's enriched right there. So you would use ultracentrifuge. Yeah, so you would take that supernatant, and then you would ultracentrifuge it, so then you would basically get sediment of the supernatant, or get supernatant of the supernatant, and then the sedimented particles are assumed to be virus. and that's called enriched supernatant. Right there, because you're ultracentrifuging it, the already centrifuged supernatant. All right, and now this was negatively stained. So a negative stain involves staining the background while the sample is not stained. In contrast, positive staining involves staining the actual sample. And staining is a technique to increase contrast in samples. In electron microscopy, electron-dense compounds of heavy metals are typically used, and contrast is the degree of differences in color that makes an object visibly distinguishable. And then these film-coated grids are a type of electron microscopy sample grid that has a thin layer coated onto its surface to provide a hydrophilic surface for the sample to adhere to, while the grids themselves are typically made of a thin metal, a metal mesh such as copper or nickel. And hydrophilic, Greek for water-loving, refers to molecules that are attracted to water and tend to be dissolved by water. All right, so yeah, this one just puts a hydrophilic surface on it, and I believe some of them put hydrophobic, water-hating, but yeah, some do this water-loving one as well. All right, so over here is a Formvar polymer, this type of polymer over there that's coated on this grid. It also has a carbon-coated grid. This is just one type of it right there for these film-coated grids. All right, let's continue further. So continuing further in the paper, human airway epithelial cells showing cytopathic effects were collected and fixed with 2% paraformaldehyde, dash 2.5% glutaraldehyde, and were then fixed with 1% osmium tetroxide, and then dehydrated with grade ethanol embedded with PON812 resin. All right, so there's a lot of technical terms right here and procedures. So glutaraldehyde is another cross-linking chemical compound, and osmium tetroxide is a heavy metal chemical compound used for fixation and or staining. And ethanol is a type of alcohol. And grade ethanol refers to a highly purified ethanol free of impurities that could affect the experimental results. And ethanol is a dehydrating agent that is able to penetrate cell membranes and replace water molecules within the cells with itself, and so on. And then this right here. Actually, I'm not sure why it says AI there. Just put delete this. So PON812 resin is an industrial resin, and then resin is a solid or highly viscous substance that is typically convertible into polymers. And viscosity is a measure of a fluid's resistance to deformation. For example, syrup has a higher viscosity than water. And embedding samples in a solid support material such as a resin is to provide structural support for the sample during further processing and imaging. Very interesting stuff here. So yes, there's just a lot of technical terms on this. So basically, they would take these human airway epithelial cell cultures, collect it, and then you would put these chemical preservatives on them, and then add this heavy metal preservative. Then dehydrate it, put alcohol on it, and then embed it in a resin or a gel-like substance, and so on. All right, very interesting. So continuing further in the paper, sections 80 nanometers were in thickness were cut from the resin block and stained with uranyl acetate and lead citrate or citrate separately. The negative stained grids and ultra-thin sections were observed under transmission electron microscopy. And note that 1 nanometer equals 10 to the negative 9 meters, or 0.001 micrometers. Yeah, so basically ultra thin. So uranyl acetate is a heavy metal stain derived from uranium. And lead citrate is a heavy metal stain derived from lead. Very interesting. All right, so now let's take a look at the summary of electron microscope preparation. So let's quickly summarize the TEM sample preparation methodology in this paper. So supernatant from the quote infected human airway cell cultures that showed cytopathic effects, aka cell membrane breakdown or death or cell death, was collected. The supernatant was quote inactivated with the chemical preservative paraformaldehyde that quote fixes or locks biomolecules in place by quote cross-linking or attaching to polymers. After 2+ hours of inactivation, the supernatant was ultracentrifuged, which involves spinning at really fast speeds of about 100,000 RPM, and this is to sediment the quote virus particles. The portion of the supernatant that contains the sedimented quote virus particles hence called enriched supernatant, was placed on thin film-coated metal grids that were stained with heavy metals, and then i.e. referred to as negative staining. Human airway cells that showed cytopathic effects were collected and fixed with more preservatives. So this was a supernatant, now it's the actual cells themselves. Yeah, human airway cells that showed cytopathic effects were collected and fixed with more chemical preservatives, fixed with a heavy metal preservative, dehydrated with alcohol, and embedded in a resin a resin block. Put a block right here. Resin block to provide structural support. And ultra-thin sections of 80 nanometers, again, extremely thin, were cut from the resin block and stained with 2 heavy metal stains in separate steps. So they would stain one and then stain the other one right after. So if we go over right here, stained with uranyl acetate and then stained with lead citrate separately. All right, the centrifuge supernatant on the negative stained grids and the human airway cells on the ultra-thin resin block sections were observed under electron microscopy. All right, very, very interesting stuff here.